In this session, I'm going to tell you about single cell protein, and thereafter I'll tell you about tissue culture, and with that I will be completing the chapter strategies for enhancement in food production. Right now, coming to the single cell protein, we know we have a problem of a very increasing population, and to supply food to this increasing population is a major concern. So we are trying to increase the food production. Of course, for that we have seen different plant breeding methods and production of different crop varieties that have a very good increase in the yield. And so now, even that is not enough, we need to search for some other sources of food. So right, we have come up with another source of food, that is the single cells. So these single cells, which are very good source of protein. So for that reason, so that's what we say, the single cell protein. Now let's look at this. Whatever that, okay, the conventional agricultural production of the cereals, pulses, vegetables, fruits, etc. So these may not be able to meet the demand of food. So why? Because of growing human and also animal population. So I mean, in case of the animal farming, we are increasing their number. At the same time, our number is also increasing, so we need to supply the enough food to humans as well as to the animals here. Now, so feeling that there is a lot of stress on these cereals, pulses and vegetable supply, even a shift from grains to meat diet. So if you like to shift, so if we say the humans to shift from grains to meat diet, then also there will be more demand for cereals. So the shift from grains to meat diet, even then what happens is it will put more demand for the cereals, because we need to supply these cereals to grow these animals. In that case, here we'll see an example that a 3 to 10 kg of grain is required to produce 1 kg of meat. Right, so to produce 1 kg of meat, we need to supply around 3 to 10 kg of the grains. So you can see that in this case also, right, only for 1 kg we require 3 to 10 kg of grains. Then definitely it puts more demand on these cereals here. Of course, this is in animal farming. Now more than 25% of human population is suffering from hunger. and malnutrition. So we need to search for an alternative source. The alternative source of proteins for both animals and humans is the SAP, that is single cell protein. So this actually what this single cell protein, so these single cell proteins are nothing but microbes. The microorganisms, some of them are edible and a very good source of proteins. Now these edible microbes, right, are being grown on on an industrial scale, right? And these SCPs, or the microbes, which are very good source of, right, proteins. Like for example, the spirulina, so which is a blue green algae. So we know the blue green algae are the cyanobacteria, right? So this spirulina, grown in large scale and will be eaten by the animals and also by humans, and actually that will be a good source of proteins. Now the point here, these microorganisms, we need not spend a lot of money to grow them, so we can actually grow them very easily. So as easy like so growing these microbes is very easy. So how means, right, you can grow these microorganisms even on materials like waste waters, can be grown on materials like wastewater from potato processing plants. So these potato processing plants, are the one which will be containing starch. So we know the potato is a very good source of right, starch, and even we can use straw, and then the molasses also can be used, which is a waste from the sugar factories, and then even you can grow them on animal manure. You can grow them on animal manure, and even in sewage waters, right? So in this case, you are actually using the waste materials from other sources, and you are growing those microorganisms on those materials. So for that reason, there is no extra amount of money you need to put and take special care for growing these microorganisms, because they will be growing very easily on these waste materials, like from these potato processing industries. That will be, those waters will be containing starch, and you can use that water and allow these microorganisms to grow in them. Then even from the straw, and then molasses, even on animal manure, and even in sewage waters, you can grow these microorganisms and use as source of proteins. Right? And by using these, you can actually produce SCP in large quantities. Can produce SCP in large quantities. Now, these SCPs, okay, the single cell proteins are rich in proteins, they are rich in minerals, fats, carbohydrates, and even vitamins. Right? So that's we say is a very good source of not only the proteins, right? They are very good source for the minerals, fats, carbohydrates, and even the vitamins here. Now, moreover, okay, so as we have seen here, we are growing these microorganisms on waste materials, and by using these waste materials in this purpose, okay, can also reduce environmental pollution. Okay, so that's I mentioned such utilization also reduces environmental pollution. So whatever that waste is, we are using it for some other purpose, and we actually helping in reducing the environmental pollution also. Certain bacterial species have high biomass production. So they have a very high biomass production, right, and even growth. So in this case, we'll take an example, a bacterium called Methylotropus. So this is a bacterium. So this bacterium has a very high biomass production as well as growth. So here you can expect from this bacterium to produce, so capable of producing 25 tons of protein, right? So that's a very huge amount of protein produced by these bacteria, Methylotropus bacterium, right? Then even we are another single cell protein examples like these edible mushrooms, okay, are now they are growing on large scale, and day by day the demand for these edible mushrooms is also increasing here. The other sources of SCP may include like this Candida utilis, Fusarium graminearum, who belong to this fungi group. Now maybe in future many microscopic fungi may also become acceptable as food to humans. This is a brief information on single cell protein. Now we shall proceed with the next one, tissue culture. The definition of tissue culture. So the technique of growing plant cells, tissues and organs on an artificial medium under aseptic conditions in the laboratory is what we call it tissue culture, right? Now, the whole tissue culture actually depends on one property that plants have is totipotency. So this plants usually in case of human beings or other animals, when they become adult, the totipotency property is almost lost, right? Now, but in case of plants, even though they become adult, most of the cells of the plant will retain this property called totipotency, right? What actually is this totipotency? So here, this is the ability of a cell to develop into a whole organism, right? So if a cell is capable of developing into a whole or a complete individual, then we have to call that as a totipotent, right? So that particular cell is capable of developing into a whole organism, like for example zygote. So zygote is a totipotent cell that has a capability to develop into a complete individual. So once, of course, now it is not possible. I cannot take a skin cell or a muscle cell and grow a complete new individual. Is not possible. It means that those skin and muscle cells are not totipotent. But in case of plants, right, even the plant becomes completely adult, there are cells, almost many of the cells in the plant will be retaining this property called totipotency, and this plant tissue culture completely depends on this property called totipotency here, right? Now we have another word for this tissue culture is micropropagation, right? So micropropagation. So tissue culture is also called micropropagation, right? So this what we mean by this micropropagation is method of producing thousands of plants through the tissue culture is what we call the micropropagation here. Now whatever those individuals which are produced through this tissue culture, right, and all those individuals will be similar to one another, or they may be completely identical, genetically identical to the actual original plant from which they have taken those cells, tissues, or any organs. So for that case, we call all those individuals which are genetically identical as soma clones. Right. So soma clones are genetically identical individuals, right? And these genetically identical individuals are almost similar, identical to their parents actually. So genetically identical individuals to their parent. By using micropropagation, we are able to grow many important food plants like the tomato, banana, apple, etc. So with the help of this micropropagation. Now let us see how this tissue culture is performed. Okay, so say suppose here we have a plant, right, and I'm going to take a small portion of this plant. So this small portion which I have taken is what I call it X plant. So now this small portion that contains cells, I'm going to grow a new plant from the cells of this X plant. I use this X plant in the tissue culture. I'm going to treat this X plant. So X plant is treated with the chlorine water of 0.1%. So this is a process what we call it sterilization. So we are going to remove any bacteria if it is present with that X plant. Otherwise those bacteria will also grow along with this X plant in the nutrient medium. So here later, once this process is completed, so I'm going to take a test tube, and that's filled with something called nutrient medium. This nutrient medium contains carbon source. So that carbon source will be like sucrose, and along with that it will also contain many inorganic salts, right? So these inorganic salts, they are also present in the nutrient medium, right? So contains carbon source like sucrose, inorganic salts, and then it also has amino acids, and it also has plant growth regulators, which I call them phytohormones, like cytokinins, auxins, etc. What I'm going to do, so this X plant, which I kept it ready by sterilization, so I'm going to transfer this X plant into the nutrient medium. So I'm going to place here, so I may take cells from this X plant, or right, so there are different types, we'll see later. So I have placed say suppose a cell here on this nutrient medium, and now as the time passes, this cell, by using this nutrient medium that contains all these different substances, it starts multiplying, and it results in the formation of many cells. So these cells, what we call them, callus. So callus consists of many undifferentiated cells. So it contains undifferentiated cells. The callus contains undifferentiated cells. Right now, later, this callus further increases, and many more cells get added, and now differentiation starts taking place, and in course of time, so here you will see a small plantlet starts developing, so inside this test tube. So this is the plant. So from this plantlet, from the callus, slowly in course of time it will develop into a plantlet here. Initiate the root and shoot formations. To initiate root and shoot formations, we need to use a high auxin to cytokinin ratio. So high auxin to cytokinin ratio. So when you are using more concentration of auxin compared to cytokinin, then it actually results in root formation. So that's what I'll say, promotes root formation. It promotes root formation. The opposite to this will be high cytokinin to auxin ratio. So this will promote shoot formation. That will promote the shoot formation. So a high auxin to cytokinin ratio will promote the root formation, whereas the high cytokinin to auxin ratio promotes the shoot formation. Right, so this is the thing, how we initiate the root and shoot formations. Then once the plantlet is formed, right, later these plantlets will be slowly transferred to the agricultural land where they will be grown and will get the yield, whatever the rest of that will be in the agricultural land. This tissue culture is also used to recover healthy plants from diseased plants, used to recover healthy plants from diseased plants, like a virus infected plants. So it's very quite difficult to get rid of the virus, but even though the plant is virus infected, you can obtain a healthy plant from those virus infected plants, right? So how is that possible? So here say suppose the virus infected plant, from this virus infected plant they are going to collect meristems. So meristems are the one who will not have the virus. So virus infected plant, from that they are going to collect meristems, especially the meristems from the apical and axillary regions. So apical meristems and axillary meristems are the one that will be free from the virus. So I'll mention here the apical meristems and also the axillary meristems will be free from virus. So hence they are going to collect these meristems and they will again place those meristems in the nutrient medium, and again that follows the callus formation, and then finally it results in the formation of a plantlet that developed from these meristems here. Now this plantlet, whatever we obtain, is free from virus, which means it is a healthy plant recovered from a diseased plant. Is already they became successful in culturing the meristems of banana, sugarcane, potato, etc. There is another process called somatic hybridization. Right. So here in somatic hybridization, what they are going to do is fuse the protoplast of two different varieties. So that is fusion of protoplast from two different varieties of plants. Means from two different species or from two different varieties, they are going to get the cells and then fuse them. Okay, now let's see here. We'll take an example of tomato. So where they have hybridized the potato and the tomato here. So it's a combination of potato and tomato. Even we have is tomato, right, where it's a combination of potato and brinjal. So like this they have tried, but whatever that offspring produced is not having all that desirable characters what the scientists actually expected. So for that purpose we are not having these still in the market. So don't search for them in the market. You are not going to get tomato and tomato in the market as they have not yet been released. So how they are going to perform this? Okay, so they are going to collect the plant cell. We know the plant cells, okay, from two different species. Okay, here we will mention that say this is from potato, okay, cell, and this is a cell from the tomato. Okay, now inside we know the cells have cell wall, okay, and there you will find the plasma membrane and all other cell organelles inside. So the nucleus, vacuole. Okay, right now in this case they are going to remove cell wall. They are going to remove the cell wall, and in this case also the same, they are going to do it, remove cell wall. Okay, and now once the cell wall is removed, so you will have only with the plasma membrane. Okay, so here I'll mention this is cell wall and this is plasma membrane. Okay, and it is left only with the plasma membrane and bounded by the plasma membrane, not by a cell wall. So now these without the cell walls are actually called protoplasts. So they are called protoplasts. Now what they are going to do, they are going to fuse these two cells. Okay, and this is a fusion. They can do it by using some chemicals or by some electrical means. We call like electrofusion or a chemofusion. So if it is in the case of a chemical use, so here I will mention in case of chemofusion, they are going to use chemicals like PEG, that stands for polyethylene glycol. So it stands for polyethylene glycol. Okay, I'll write it again. I think it's not. So in short, it is called PEG. So by using such chemicals, they are going to fuse these two protoplasts, and once the two protoplasts are fused, you are going to get a hybrid here. Okay, So you are going to get a hybrid, and this one is actually, you call it the somatic hybrid. Called a somatic hybrid. Okay, now this somatic hybrid is allowed to grow into a new plant. Okay, of course with the same tissue culture process, and they like to see whether the characters of this potato and tomato are seen in this somatic hybrid. So of course unfortunately they did not find, and because of that these are not available in the market. This is all about the somatic hybridization, and with this I complete this tissue culture and also the strategies for enhancement in food production.